We're going to have 3 more trainee presentations. And our first one is a presentation from Dr. Basit Yousuf, who works at Canadian Blood Services head office in Ottawa. And I will put your slides up. Wait a sec, I better share first. And Basit, I'm assuming you're out there.
Yeah, I'm here. Thank you.
Okay, great. Okay, so there are your slides. And I can drive them for you. Go right ahead.
Okay, can I start?
Please do.
Okay. Hello, everyone. My name is Basit Yousuf. I am working as a postdoctoral fellow with Dr. Sandra Ramirez at Canadian Blood Services. I am presenting my today's talk on enhanced capsule and biofilm formation in Staphylococcus aureus grown in platelet concentrates as revealed by transcriptomic analysis. Next slide, please. Platelet concentrates represent an essential medicine. I'll be referring them as PCs. And PCs are prepared in well-aerated PVC bags in the presence of high glucose concentration and are kept under constant agitation for up to 7 days at room temperature. These conditions are required for maintaining the maximum functionality of platelets. However, these settings make PCs vulnerable to bacterial proliferation. Staphylococcus aureus, an opportunistic pathogen, has been reported as one of the predominant bacterial contaminants in PCs. It is introduced into PCs during venipuncture at the time of blood collection, as it commonly resides on human skin. Although PCs are strictly screened for bacterial contamination before being transfused to the patients with back alert culture system, however, Staph aureus has the ability to escape PC screening detection and cause septic transfusion reactions. Next slide, please. So our research question was to unravel the Staph aureus factors involved in escaping PC screening detection and immune response evasion. In order to address these questions, we spiked PCs and tryptase soya broth with 2 Staph aureus strands, CBS2016 and PS3117. CBS2016 escaped PC screening detection and was isolated post-transfusion after causing a septic reaction, whereas PS3117 was detected during PC screening. These 2 strands were allowed to grow up to stationary phase. A Staph aureus RNA was extracted and sequenced. Differential gene expression between PC and TSB conditions was determined. Next slide, please. Results revealed significant increase in the transcription of CAP, A, B, C, as you can see in the above column. And these 3 genes are vital for capsule biosynthesis. Capsule biosynthesis is encoded by an operon, which includes 16 genes from CAP A to CAP P. And these genes had more than 4-fold upregulation in PCs. Moreover, the positive regulators of Staph aureus virulence, positive regulators of capsule biosynthesis, were upregulated in CBS2016 also, which include transcription factors like SIG-A, SIG-B, SPOVG, whereas in PS3117, they were downregulated. Also, the 2-component signaling system, ARLRS, which is positively regulating capsule biosynthesis, was upregulated, whereas PSI-RS, which is a negative regulator for capsule biosynthesis, was downregulated. Capsule plays an important role in the development of invasive disease and immune response evasion, and is also a potential vaccine candidate. Next slide, please. Moreover, we observed the Accessory gene regulator, the master regulator of Staph aureus virulence, which controls the production of numerous virulence factors through AGRA and RNA third. We observed strong repression in phenol-soluble modulin, RNA third, and proteases, which negatively control the biofilm formation, whereas the surface proteins like clumping factor A, B, and protein A, which positively control the biofilm formation, were highly upregulated in CBS2016 strand. Biofilm formation plays an important role in surface proteins play an important role in platelet activation and aggregation, as well as evasion of immune response. Next slide, please. Additionally, at the metabolic level, we observed a strong upregulation in the anaerobic metabolism genes, particularly pyruvate formate lyase, formate dehydrogenase, and acetyltransferase. These genes play an important role in the biosynthesis of proteins, DNA, and RNA. and under anaerobic conditions. Also, the anaerobic ribonucleoside reductases, which are involved in DNA synthesis and repair under anaerobic conditions, were highly upregulated in CBS2016 strain and less in PS3117. Notably, aerobic ribonucleoside reductases were upregulated only in PS3117 strain. Currently, we are working on validation of these results using RT-PCR and Western blotting and mutational studies. From these results, we concluded that CBS2016, the strain escaping PC screening detection, had an adaptive mechanism to strongly enhance capsule and biofilm formation and shift towards anaerobic metabolism, possibly contributing to its enhanced infection development, immune response evasion, and misdetection. These results were strain specific, as we didn't observe these results in the PS3117. So I thank you all, and I'm open to the questions.
Great, thank you very much. So I will stop sharing my screen so we can get everybody's smiling faces back up here. You can either put a question in the chat box or raise your hand and ask a question directly. So I'll just ask a question. So
yeah,
the
please.
way that we screen for bacteria and platelet concentrates is a culture-based method. So why do you think the bacteria just weren't growing, weren't dividing very rapidly in the culture bottle, or we didn't catch one when we sampled, or why do you think we missed the
actually
that strain?
that was actually the question, why the bacterial strands, or some bacterial strands, are missing in the back alert culture system. So there are different reasons from what we observed as of now. First of all, the strand which I was working on, it grows slow compared to the other strands. And also these are the factors like biofilm formation and the anaerobic metabolism, which we observed through the transcriptome analysis. Because in anaerobic metabolism, we have the less carbon dioxide production, and the back alert system detects the carbon dioxide released during the metabolism. So it may be one of the reasons. And of course, the slow growth is also the reason.
Okay, thank you.
Yeah, you're
other
welcome.
questions? I know there's some microbiologists out there. Okay, well, it looks like there aren't other questions, but thank you very much, Basit. That's an interesting project.
Yeah, thank you so much.